Good morning, everyone. This is the educational session, our first session of ESHG 2020. The first time as a virtual conference. We are very happy to kick off the conference of this educational session, where we have 4 great speakers. You're going to notice that we don't have all 4 speakers live with us this morning. We only got 2 of them because the others are at least 6 hours away in time. So Jonathan Weissman and Doug Fowler, we don't gonna have questions, unfortunately. Please feel free to contact them by email. And for Alex Hus1 and Matthew Loser, we're gonna have questions here from you. Write them in the chat, and we're gonna ask them to the 2 speakers. So like I said, our first speaker is going to be Jonathan Weissman. I think he, from what I saw from his slides, he's going to briefly introduce his affiliation and that he just recently transitioned to a new position. So I'm just going to mention briefly, he's a very well-established scientist with more than 200 peer review. Like I said, our first speaker is going to be Jonathan. Why am I muted by the host? Sorry. I'm back. I'm very sorry. So he has developed ribosome profiling, and he also developed a lot of CRISPR assays, including the dCas9 version of CRISPR to target CRISPR to DNA without cutting. We're very much looking forward to his talk.
Good morning. My name is Jonathan Weissman, and until very recently, I was at UCSF in San Francisco, and my lab has now moved to the Whitehead Institute at MIT. So it's a pleasure to be able to join you virtually, although I would very much like to have been there in person. I'm going to tell you today about our efforts to use ultra-rich phenotypes using single-cell RNA-seq to map out and understand how a combination and expression of genes leads to complex phenotypes. One of the major surprises, I think, from genomics and single-cell approaches has been the lack of relationship between the number of genes and the complexity of an organism. So a simple organism like Saccharomyces cerevisiae has 6,000 genes, C. elegans have 16 or so, and humans have 20,000. And on top of that, we now know that there are up to a few thousand distinct cell types from single-cell RNA-seq approaches. So the question then arises, how is it that you get such a large number and diversity of cell types from a small number of parts? And of course, the answer is combinatorics. So in the same way that you can have a simple keyboard, piano keyboard with 88 keys, and by changing the cadence of the keys and the order in which you hit the keys, you can play anything from Mozart to Ray Charles. The combination of these relatively small number of parts can give you the enormous diversity of metazoan life. And for now coming on 20 plus years, thanks to first microarrays and then RNA-seq and a whole range of different approaches for monitoring gene expression, we've been able, as it were, to listen to this music, to understand under different conditions, different differentiation or physiologic states, what the set of genes that are being expressed and how strongly they're being expressed. But until relatively recently, we were left at this sort of observational stage. But now, thanks to CRISPR, we can actually essentially compose new music. We can change the expression of the genes and see how this affects the physiology and the state of a cell. And the original nuclease, while a powerful tool, essentially would let you break fix a working key or fix a broken key, but not sort of allow you to change the expression in an analog way to single or multiple combinations of genes. For that, slate modification on this tool really was the most powerful approach. And that was using a catalytically dead Cas9, so basically breaking the scissors of Cas9. And what this did is allowed one to fuse on either transcriptional inhibitors and therefore in a programmed way shut down or lower the expression of neighboring genes or transcriptional activators and allow you to turn on the neighboring gene. And this is work that was started really at the dawn of the modern CRISPR era at UCSF in a wonderful collaboration between Stanley Chi, who was then a fellow, my lab, Wendell Lim's lab, and Jennifer Doudness. In essence, what we had is a volume control for each gene or combinations of genes. And so with that, I'll get back to this idea of combinations. And other important concept to introduce is the notion of a genetic interaction. And essentially, a genetic interaction is asking, does altering the expression of a combination of genes allow or give you a phenotype that you couldn't predict from the individual effects on their own? And so that can be thought of sort of most simply by gene knockouts and say a simple readout like fitness, where you might expect that if you knock out one gene A, you get a cost of fitness, and gene B, you get a cost of fitness. And then the question is, what happens when you do the combination? And for most genes, they have no interaction, and the double is a little bit sicker than the single, and there's sort of a way of predicting on average what you expect. But for some special combinations, you get so-called buffering interaction, where, for example, once you lose gene B, there's no further cost to losing gene A. And then for a difference, and this would be what you'd expect, for example, if you had a parallel pathway of A producing an intermediate that went to B, and then the product. So if you've lost A or B, you lose the entire pathway. Alternatively, there's subsets of combinations that give you synthetic lethal interactions. And this is, for example, what you'd expect if A or B individually contribute to an essential process, for example, making ATP. And so as long as you have one or the other, the cells are more or less okay, but the combination is lethal. And there's been a great deal of interest in mapping these types of genetic interactions. And really much of the groundwork for this has been done in the Saccharomyces cerevisiae in large part pioneering work from Charlie Boone, as well as from my lab and Nevin Krogan and a number of other groups. And from this, it's been clear that this is a powerful approach for allowing the principal definition of gene function it can also identify suppressors, for example, in genetic diseases where you'd have inherent loss of one gene, and you'd ask, is there some other gene whose expression you could change that would help modify or ameliorate the effect of that loss? And then finally, it's a strategy for enabling rational searches for combination therapies. And this is most relevant perhaps in the cancer field, where cancer might have a loss of function, say, for example, a BRCA gene, and then by finding other genes that you could lose that would create a synthetic lethality, it's a way of finding a selective vulnerability. And of course, this is the basis for successful therapies with the PARP inhibitors, which show a strong synthetic lethal interaction with cells that have lost the BRCA gene. So as I mentioned, there's a sort of a long history predominantly in Saccharomyces cerevisiae, but in a number of other sort of small model organisms. But with the dawn of CRISPR, it now became not only possible, but really feasible on a very large scale to start mapping out these genetic interactions in human cells. And we and others in the last couple of years or so have begun to do this in large scale. And in particular, in a wonderful collaboration with Luke Gilbert, my UCSF colleague, we basically took advantage of the fact that you could, by a simple ligation and fusion, create a massive double library. So if you have 1,000 guides that are targeting each of 1,000 different genes. By ligation, you can create a library with 1 million gene pairs. And now if you put each of these pairs of guides individually in a cell and then do a competition experiment, and read out by next-gen sequencing in this massively parallel experiment, you can measure the relative growth effects of these million or so combinations, and then go through this exercise of saying what is the growth effect of the double knockdown, and how does that compare to the single, and where and in what cases you are getting this type of suppressor or synthetic lethal interactions that we are interested in. And this work is published, so I'm not going to go into it in too much detail, but basically the outcome of these is these massive and really enormously both complex and I think biologically rich maps in which basically the x-axis would be the first gene knockdown, the y the second, so it would be 1,000 by 1,000, and each spot is a measure of the inferred value to the extent to which there is a genetic interaction, buffering, or synergistic. And as you sort of load these up in order genes rather by their similarity in their paths of, in their patterns of genetic interactions, it's a powerful way, as I alluded to earlier, of identifying genes that have a similar function without having a priori to postulate what those genes might be doing. So very nice principled way of getting at gene function. And here's an example of this where we have 4 NDUF genes involved in the oxidative phosphorylation, and then at that time on characterized gene TMEM261, which had a virtually indistinguishable pattern of genetic interactions, and in fact we were able to show that it has the same phenotype and is acting directly with together with NDUF to promote oxidative phosphorylation. And more globally, we were able to show that we can find many more new such interactions, the majority of which were not in the existing databases like the string databases. So again, as I said, sort of a nice way of identifying gene functions, genes that interact with each other in a principled and systematic manner. But none of this really gets at why 2 genes, let's say gene A and gene B, have a synthetic interaction with each other. And that can sometimes be rather obscure because it gets at the underlying mechanisms by which the cell is wired together. So we may be able to measure that loss of A and loss of B causes synthetic lethal interaction, but understanding where that comes from is much more challenging, and I'm going to illustrate that in the next slide or so. And that's really, understanding this is really going to be the rest of the focus for the rest of the talk. And I would say it's important to understand this because if one, it really tells you something about how the cells wire together, and 2, from a very practical point of view, if we're going to start exploiting these genetic interactions or synthetic interactions to, for example, to get combination of drug treatments for cancers, we really want to understand where they're coming from. So I'm now going to focus on one of these genetic interactions. And I told you we did 1,000 by 1,000, so there were 1 million such combinations. This one was a striking one, admittedly, but not entirely atypical. So this genetic interaction was between 2 genes, one called FDPS, the other one called Hus1. And really what you should remember from this is that these 2 genes would not be expected to have synthetic interaction. They're very well characterized, and they're involved in quite different biological processes. So Hus1 is involved in the 911 complex that recognizes and responds to DNA repair, especially replicative DNA damage. And FDPS is a key enzyme in cholesterol biosynthesis. So you might think at first, perhaps lack of cholesterol was in some way causing DNA damage, but we knew that wasn't the case because actually, if you added a statin or genetically ablated upstream the cholesterol biosynthesis pathway, you'd get rid of the synthetic interaction. So to make a long story short, what we were able to piece together with a lot of directed and experiments that, as you expect, took us several months just to track down this one genetic interaction. We found that there was an intermediate IPP that was accumulating when you knock down FDPS, and this intermediate basically was leading to either directly or indirectly inhibition of ribonucleotide reductase that was causing a depletion of deoxynucleotides. The lack of DNTPs was causing replicative damage, and the cell did okay as long as you had the Hus1 911 system for sensing and responding to this damage. But if you had this toxic intermediate interfering with DNA replication, and you lost the 911 response, then cells were unable to survive. More generally, I think I went into this example in a little bit of detail because I think it illustrates both the challenges and opportunities for understanding how combinations of genes or change in expression of genes lead to rich phenotypes. And really, we can think of the state of the cell as being a rich, high-dimensional phenotype that sort of moves along a manifold, which describes the transcriptional state or the physiologic state of the cell. And as we add perturbations, we're changing the position on this manifold. And this is sort of analogous to Waddington canals that many people may be familiar with. And we can do a separate set of perturbations, and it will move it to an entirely different place on this manifold, this high-dimensional space describing the state of the cell. Now, if we do essentially a dimensionality reduction, looking only at fitness as the outcome, these two very different states, state A and state B, could have exactly the same or indistinguishable fitness effects. And that's basically because when we look at fitness, that's a very crude readout, and there's many, many different ways to being unfit. And that really underscores the challenges of just doing simple growth-based genetic interaction maps. That you essentially, when you go through this dimensionality reduction and look only through the lens of fitness, you lose information that you simply can't retrieve after the fact. So more broadly, the challenges with these one-dimensional phenotype-based genetic interaction maps, as powerful as they have been in both model organisms and now in human cells, is that first, fitness is broad, but there's many ways to be unfit. You can have the cells die, they can have slowed growth, they can have cell cycle arrest, they can terminally differentiate. So we need richer phenotypes to distinguish between these possibilities. And then on top of this, there's the issue that we're looking on an ensemble average of the cells, and that can obscure the underlying causes of or roots of the genetic interaction. So I'm going to take a little bit of diversion for one slide. It perhaps is not entirely relevant, and I borrowed it from a wonderful former student of mine, Silvia Ruskin, who's now a Whitehead Fellow. But I'm going to show it anyway because probably if you remember nothing else from this talk, this is the thing that you will remember. So it's really illustrating the problem with this of population averages. So 1985, or actually the class of 85 from UNC, the average starting salary for the geography majors graduates was about $130,000. And you might think UNC is a good school. Geography doesn't seem like the most modern major, but who knows? But then when you look at the data, you see that there were actually 9 students graduating from the class of 85. 8 of them earned just north of $10,000, and 1 earned more than $1 million, and that one was Michael Jordan. So, just the point being, if you were to take a naive average of these 9 students, you get a nonsensical, misleading answer. In the same way, each cell acts and lives on its own, perhaps communicating with other cells, and if you start to take averages, you can really obscure and lose information. And until fairly recently, that was pretty much what we had to live with. And But what's really changed has been the sort of explosion of single-cell RNA-seq technologies. And I'm going to focus on approach by 10x, which allows you to do sort of hundreds of thousands of cells at once, but there are a number of different technologies. And a few years ago, my lab, in collaboration with Aviv Regev, developed an approach called Perturb-seq that lets you take advantage of this single-cell resolution to understand how perturbations change the activity of a cell. And I should say that there were a few other approaches that came out at about the same time, and to rough approximation, they perform equally. And so the basic idea, and I'll focus on the Perturb-seq implementation, is that you would put in each cell a different guide RNA that would cause a different gene to be either knocked out or knocked down or overexpressed, or in the case of genetic interactions, combinations of genes, and then run it through a 10x single-cell RNA-seq, and now each cell, the mRNAs from each cell would be uniquely labeled by a barcode, so that let you analyze the transcriptional response in that cell. In addition, we were able to capture the information of the guide in that cell. So this allowed us basically to, for each cell, map what was the perturbation, which gene or combinations of genes were changed, and then how was this perturbation changing the state of the cell, pushing it on, in this manifold analogy, pushing it along the manifold. And I'll illustrate this, how this was helpful for interpreting genetic interactions, coming back to this FDPS and Hus1 example. And remember, I was telling you that FDPS was causing a toxic intermediate, which was causing DNA damage that was being sensed by the Hus1 911 DNA damage response. And we can see this here, where by looking at the distribution along the cell cycle of cells that either weren't perturbed, were perturbed with Hus1, which, because it's essentially a sensor, as long as there's not some challenge, does very little to change the cell cycle. But if we perturb FDPS1 and cause this toxic intermediate, we now accumulate cells in S phase because there's replicative damage and the cells are stopping until they repair this. But now when we do the double, we see basically the cells blow through this S phase, despite the fact that there still is a replicative damage, and end up in G2M, and the cells that are in G2M are the ones that are actually dying. So we get really, I hope this illustrates how by having this richer phenotype, we're able to understand and have a much more in-depth mechanistic understanding of what the basis of this synthetic lethal interaction was. More generally, we can take a type of genetic interaction map that I showed you previously and pick combinations of genes like FDPS and Hus1 that give you these unique synthetic or buffering interactions, and then pick these combinations and do a large-scale rich phenotyping via Perturb-seq and map where along this manifold these combinations are and how that differs from where they're expected. And the beauty of this sort of approach is that instead of relying on the dimensionality reduction of growth, which is completely uncontrolled and really kind of obscure to unpack after the fact, there's nice mathematical approaches for doing projections into two dimensions, like UMAP or TISNE approaches that retain as much of the structure as is possible. So again, the idea is we pick sets of, we do large-scale growth-based genetic interactions. We pick combinations that are giving us interesting combination genes that give us interesting genetic interactions. Use the Perturb-seq and mapping on the manifold to understand these. And of course, if this wasn't generally useful, I wouldn't have taken you this far. So here's the example of a few hundred genetic interactions, and this is in a K562, an erythroid progenitor cell. And what we see is just how many different rich and interesting ways there are for the cell to lose fitness. So for a subset of these interactions, it's just inducing apoptosis, the cells are dying. But for others, it's causing a arrest in the cell cycle. And then for others, it's causing differentiation into, for example, megakaryocyte, a granulocyte-like state, and an erythroid-like state. So if we're thinking now back at the sort of synthetic lethality as a potential treatment, this shows how you can use perturb combinations of genes to either specifically kill this cancer cell, or alternatively to cause it to terminally differentiate into, in this case, at least one of three distinct states. So I told you that there was this challenge with genetic interactions based on growth, that simple measurements conflate very different outcomes. But the second challenge is that there's just a huge scale. If we're talking about 20,000 genes, and there's 20,000 squared over 2, or 200 million possible combinations. And while that might be possible to do once or twice by growth, it certainly is impossible to do by the full Perturb-seq approach. And in any case, it really is impractical if one wants to map out genetic interactions in a variety of different cell types or more physiologic conditions, either an in vivo or an organoid type system. But we also know that genetic interactions are uncommon. That's what makes them so valuable. Most genes don't interact with each other. And this essentially mathematically means that we have a low rank matrix. And there's fortunately, really nice mathematical approaches for how better to sample when you have a matrix like this that has low rank or is sparse. Most of the interactions aren't interesting. And I should say, Aviv Regev has really done beautiful work on so-called compressed sensing type approaches for measuring, for making more efficient or more thoughtful ways of mapping out these types of genetic interactions. And I'm going to talk about a different approach that was developed by Tom Norman, who was a postdoc in my lab and recently took an independent position at Memorial Sloan Kettering. And the approach he used was a so-called recommender system, or you might be more familiar with it as the Netflix problem. And the idea is you'd have side information. So here in the Netflix manifestation, you were talking about how do you predict, or if you know the demographics of a user and you know something about the movies they like, how can you predict which other movies they like? And the idea is you'd have side information about the users, you know, what their age, sex, demographic is. And then you'd have side information about the movies. You would then, you know, categorize movies by, are they slasher movies or rom-coms, for example. And that if you have a lot of information about which users like which type of movies, and you know some empirical data about your particular user, you can actually do quite a good job of predicting which movies a given user would like. And again, this is possible because it's a sparse matrix. Most people don't like most movies, so you're trying to find the sort of rare gems. And in the same way that you can use side information about users and about the movies, we can have side information about gene A and side information about gene B. In this case, our side information is the individual perturbation or Perturb-seq experiments for gene A. So if you alter gene A, what is the change in transcription? If you alter gene B, what is the change in transcription? And then if we have some empirical information about which genes tend to show genetic interactions, we can then use this recommender system to predict new interactions. I'll just give you an example of this. Here's an actual genetic interaction map in which we've measured empirically the growth effects and the genetic interactions between each of the combinations. We can then randomly downsample this, so we're only using 10% of the fitness data, and then use the recommender system basically to really very nicely fill out this type of information. So I think more generally, this combination of the really dramatic advances in the scale in which we can measure genetic contractions, both by growth and by single-cell RNA-seq, together with these sort of more thoughtful experimental approaches that let you sample this space in a much more efficient way, really hold promise to dramatically increasing our ability to map out these types of genetic contractions, basically to map out how combinations of genes give you unique, interesting, and non-obvious phenotypes. Getting back to this original question that I posed is, how do you get such rich behavior from a small number of parts? And I would also argue that this really has very broad implications for a wide range of biological problems, including human genetics. So from developmental, just an example, from developmental, and for development, the interactors would be different transcription factors, and you would then want to understand which combinations would give you a different cell types. Basically, doing the Yamanaka experiment, but in a much larger scale, a much more thorough and systematic way to very efficiently map out how you can do combinations of perturbations or overexpressions of genes to give you a given cell type. From a cancer point of view, we would like to understand from mutation in genes that are essential for the cancer or integral to the cancer, what are the synthetic lethal interactions? So this is sort of the perspective that you can think of the analogy that cells a stool with 4 legs, and when you lose BRCA, you lose one of them. It's still stable, but you want to understand which is the next one you can pull out that will specifically make those BRCA minus cells unviable. And then from the point of view of genetic diseases, we want to understand how the SNPs or genetic variations can combine to give you variable expressivity of disease. So I've talked, I hope I've convinced you that by having these rich phenotypes, in this case in the form of a transcriptional response, a single cell RNA-seq responses, that we're able to understand and map out how combinations of genes push the cell essentially along this manifold to give you interesting phenotypes. But of course, transcription is only one type of rich phenotype, and this basic formalism that I've talked about could be generalized to many different types of rich phenotypes, and the experimental approaches are now really starting to catch up with that, including some really beautiful posters from Zhuang and Paul Blaney that are now enabling, and others that are now enabling large-scale pooled optical screens. And so I'll just finish with a plug for a different approach. So we were very interested in this space and of DNA damage and DNA repair, and the outcomes from, for example, when you have a CRISPR-Cas9 cut, which can give you a wide range of different insertions and deletions, and using the next-gen assay, next-gen sequencing assay, Jeff Hus1 and Britt Adamson, who recently left my lab and is now assistant professor at Princeton, have used this to map out how alterations in individual or combination of DNA repair pathways lead to change the different outcomes of these repair events. So really mapping out in a much more principled way, understanding how DNA, the combinations of DNA damage factors, the balance of different factors lead to different repairs, and that we think will be both important for fundamental understanding of DNA repair, but also from a very practical point of view of trying to control repairs of Cas9, either for research or for therapeutic efforts. So with that, I'll thank you, and of course, the most important slide, thank the people who did this work. The Perturb-seq approach was a wonderful collaboration with Aviv Regev and Trey Dixon and Oren Parnas in her lab. And from my lab, it was really led by Tom Norman and Britt Adamson. And much of the genetic interaction manifold work has been just a terrific collaboration with Tom, Luke, and Max. And I should say, Tom is now, as I mentioned, at Memorial Sloan Kettering. Luke is assistant professor at UCSF, and Max has moved on fairly recently to, he's an MD PhD back in the clinic. And then the DNA repair work was a terrific collaboration between Jeff, Britt, and Cecilia Cota Romanosino and Bothner, who were at the time at Editas. So thank you, and
going on air.
Okay. Good morning. My name is Malte Spielmann, and I'm not sure what's happening. The presentation is already starting, but I would like to, first of all, welcome you again, also from my side, and introduce the next speaker. The next speaker is Matthew Lose, as you can see, from the University of Nottingham. And since we're in the technology session, we are changing gears a little bit and going really into sequencing, I think very relevant to everyone listening out there. And we're going to hear about nanopore sequencing. And I'm very excited, at least from what I understood, lots has happened in the last year. So take it away, Matt. Thank you. And just to finish this off, he will be available for questions after the talk, and you can post also you can post the questions in the chat, and then we will give them to Matthew, and he will answer them. Thank you.